On the cover of Science Times this week, a startling look inside the brain. Edvard and May-Britt Moser are neuroscientists whose discoveries are changing the way we think about how we remember where we've been. They and their work are profiled in Science Times by reporter James Gorman, and they join us now from their laboratory in Trondheim, Norway. Hello.
Hi.
Thank you very much for joining us. Can you talk about a little bit about your research and what you have learned?
Yeah, I don't know. I could start and maybe Edvard could follow up, but at least it's quite exciting to understand how behavior is created by the brain. And then since we wanted to study rodents or rats, then to study spatial navigation is very easy. And then we knew about the work by John O'Keefe in London, and we also collaborated, or we even went to him, and he was supervising us in how to study spatial navigation and studying single cells in the brain and how they were related to spatial navigation.
Yeah, so what John O'Keefe discovered already in 1971 was that there is a type of cell in the brain that is active only when you are in certain places in the room. And those cells were called place cells. And what we wanted to find out, and which we have worked on when we started our own lab, is how do these cells relate to other cells in the brain? Are there other types of cells that also code for space? And how do they all work together? And we're trying to use that as a way to understand how the brain or the cortex of the brain is working in general, trying to find general principles. And the advantage we have is that these types of cells, as May-Britt says, are really easy to measure. So you can actually use them as a window into the deep parts of the brain.
Mm.
Talk a little bit about the animals in which you do your research.
Yeah, so we're using mostly rats, also a little bit mice. And what we are able to do is to use very thin sensors that are implanted into the brain so that we can actually almost navigate these sensors between the cells and then listen to their natural activity, pick that up and then amplify it some 10,000 times. And then we can actually see on an oscilloscope the activity of individual neurons when the animal is finding its way through the environment.
And not only see, but we can even hear it. And the sound is like music in my ears because it's like popcorn. So whenever the rat is outside the place field in an environment, then the cell is silent. But when it starts to go into a field, then you hear pop, 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 pop. And then you go into the field and then the pop, 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 pop. Like you put a bag with popcorn in the microwave oven and you start to hear now it's pop, pop, pop. And then it's so frequent that you have to take the popcorn bag out. Then you know you are in the middle of the place field in the environment.
One of your key discoveries involves something called the grid cell. And I'd like to ask you to talk about what is a grid cell and what does it have to do with our memory of where we are in space?
So grid cells are cells that are active only in certain places in the room where you are, but many places. And the fascinating thing is that those different places where a cell is active form a very regular hexagonal pattern, so that it consists of almost like triangles, equilateral triangles that repeat over and over again. So it's almost like a matrix or like a graphics paper that you put over the room, and then the cell fires only at the cross sections. But the difference from graphics paper is that it consists of triangles that then repeat over and over again. And this kind of map, it's almost like intersecting lines. It contains very accurate information about where you are in the environment in meters and angles, so distances and directions. And we know that this information is there, and it's very likely that the brain actually then uses it also to measure where you are in space.
Where do you see your research leading? Will it help in diagnosing and treating disorders of memory like Alzheimer's?
Yeah, what is exciting is that when we found these grid cells, we found them in a place in the brain that is called the entorhinal cortex, which is connected highly to another place in the brain that is called the hippocampus. And if you lose the hippocampus, you can't get new types of memories about events and facts. And if you have a tiny damage in the entorhinal cortex, you can't find your way. And you can't find your way if you have a damage in the hippocampus also. What they have shown is that there are some groups of Alzheimer's patients that have a tiny lesion in the entorhinal cortex, exactly where we found the grid cells. And if they start to see this lesion, they can say whether this person is going to develop Alzheimer's disease or not. And the first symptom these patients typically have is that they can't find their way in the environment. So the spatial navigation system is out of function.
So very important work you're doing. Edvard and May-Britt Moser lead the Kavli Institute for Systems Neuroscience in Trondheim, Norway. I want to thank you both for coming on the podcast and telling us about your research.
Thank you.